Hi everyone. My name is Philip Babby, and I'm super excited today to talk about the effects of these nanoplastics, low-density polyethylene, on methanogenic communities in paddy fields in China. First, let's go through the trouble of defining the importance of this research. Obviously, number one, rice is one of, if not the most popular foods in the world, depending on your source. Rice is used as 10% of global arable land and is also the cause of 10% of global methane emissions. Our current understanding of the interactions of plastics with these fields is important to maintain global supply chains and global health, as plastics are commonly used to maintain rice farming. The main question we want to ask is how these plastics will interact with microbial communities in rice fields, and as research has found, there are significant interactions between the nanoplastic particles that come from rice farming maintenance and methanogens that are responsible for this huge methane production. Currently, our research questions pertain to the long-term effects of micros and nanoplastics on the environment. Whether these plastics have unique interactions with microbial organisms due to their size and adsorb and absorbability, whether these interactions are positive or negative, the types of organisms impacted by these interactions, as well as the implications related to waterlogged rice paddies and rice farming, as well as other crop farming on a global scale. Let's begin about talking about the issue. Anthropogenic emissions are a global concern that encompass a wide variety of processes and originate from a multitude of sources. Examples could be greenhouse gases, wastewater, nuclear waste, and many others. The long-term effects of many of these processes and byproducts are poorly understood and still undergoing research. Plastics, among many of these byproducts and products themselves, are among some of the most widely produced and discarded emissions, as well as their highly diverse nature that contributes to a unique chemical composition and subsequently its reactivity. By definition, microplastics are plastics that range up to about 1 micrometer in size. And subsequently, nanoplastics are plastics defined as anything under that threshold. Most of these plastics are byproducts of hydrolytic reactions with larger macromolecules, as well as UV damage from the sun, and can still be influenced by a wide range of anthropogenic byproducts and chemical reactions. They're able to be absorbed by a wide variety of organisms, including microbes and macroorganisms. They are extremely widespread. An estimated 18.1 million microplastic particles per hectare are accumulated in Chinese soils per year. Many of these soil micro and nanoplastics in Chinese rice paddies comes from film mulching techniques, as shown on the screen here or here. Low-density polyethylene, shortened as LDPE, is an ethylene homopolymer that engages in high branching activity. It breaks down easily because of its low density, which is about 0.91 grams per centimeter cubed, and it is used widely in commercial products and packaging. Its size is usually a result of coagulation of nanoparticles. In this case, we usually see about 50 nanometer particle sizes. Our concentrations that we find naturally in rice soils usually range between 0.0% and 6.7% in Chinese rice paddies. An estimated 52.8 million tons of this plastic are produced annually globally. Let's define methanogens. Any organism that synthesizes methane in processes related to cellular metabolism is a methanogen. These are typically associated with archaeal species. However, some fungi and bacteria have also been shown capable. There are 3 known pathways that all utilize different carbon substrates: acetoclastic, which uses acetone; methyllotrophic, which uses methanol or methylamines; and hydrogenotrophic, which utilizes carbon dioxide and hydrogen gas. Methanogenesis primarily takes place in anoxic conditions. However, controversial novel research is currently being undergone describing aerobic methanogenesis. 2 soil samples are utilized in this experiment. Black soil from Jianzhenjian Agricultural Experimental Station in Heilongjiang, its pH is about 6.64. Total nitrogen concentration is about 1.64. Soil organic carbon concentration is 15.78, and carbon-nitrogen ratio is 9.64. Our other soil is red soil from Changsha China National Rice Institute in Changsha in Hunan. Its pH is about 5.96. Its total nitrogen concentration is 1.97. Soil organic carbon concentration is 10.26, and the carbon-nitrogen ratio is 10.38. Now let's begin to talk about methods and materials used in this research. Let's begin talking about the microcosms. We have 2 soil samples, black and red, along with 4 concentrations that will be used per soil sample, representing the total carbon concentration. We have 2 time sampling points, representing the first 30 days, which is usually the amount of time that hydrolytic decomposition takes in anaerobic incubation of polymer substances like LDPE, and 160 days to measure long-lasting effects. We also have 4 replicants of each microcosm, representing 64 total microcosms. Preparation of these microcosms included 20 grams of soil added to 40 milliliters of autoclaved water, mixed with their appropriate nanoplastic concentration. Each sample was sealed and flushed with nitrogen gas for about 10 minutes to create anoxic conditions. All samples were incubated for 25 degrees Celsius for 160 days. Sample measurements. 100 microliter samples were taken from the headspace of microcosm chambers using VC pressure-locked syringes. These gas emissions were then measured via gas chromatography. Dissolved organic carbon, shortened as DOC, were analyzed in Landgesellbohl, Germany by Elementar using a total organic carbon analyzer. The calculated carbon dioxide values were not fully representative of released carbon, as biochemical pathways in methanogenesis also allocate carbon uptake into cell biomass. Solid phase extraction was responsible for extraction of this dissolved organic matter, shortened as DOM, from the control and the 0.5% treatments using Fourier transform ion cyclotron resonance. Let's talk about the targeted metabolomics. A 2 milliliter liquid sample of each microcosm was centrifuged for 15 minutes at about 18,000 Gs at 4 degrees Celsius. Short-chain fatty acids, shortened as SCFAs, were analyzed using an HP 6890 gas chromatograph. Another 2 milliliter liquid sample from each microcosm was homogenized with a 300 microliter isopropanol acetonitrile 1:1 solution, and subsequently centrifuged at 18,000 Gs for another 10 minutes. The supernatant was then subjected to long-chain fatty acid, LCFA, analysis using UHPLC. DNA and RNA extraction in qPCR. DNA was extracted using PowerSoil DNA isolation kit from a 0.25 gram sample of wet soil. Purity and concentration of DNA extracts were verified by nanodrop spectrophotometry, and extracts were subjected to a negative 20 degree Celsius storage until qPCR and metagenomic analysis. RNA was extracted using 0.5 grams of wet soil sample mixed with glass beads in a TPM buffer. It was then subject to a vortex and centrifugation at 20,000 Gs for 5 minutes. Supernatant was extracted with phenylchloroform isoamyl alcohol and chloroform isoamyl alcohol. RNA was precipitated with isopropanol and resuspended in a 50 microliter TE buffer. Genomic DNA was removed using turbo DNA-free kit, and total RNA was concentrated and purified using RNeasy Minolute kit. MCRA stands for methyl coenzyme M reductase. It catalyzes the final step in methanogenesis of all pathways and is present in all methanogens. The number of these genes were determined using qPCR, with all qPCR reactions performed in 3 biological replicates with 3 technical replicates. qPCR was carried out using a CFX Connect real-time PCR detection system. Like RNA sequencing, qPCR and real-time qPCR of mCRA were each performed from 30 samples. The PCR efficiency was 89%. This slide seeks to show the workflow process of a metagenomics library creation. 36 metagenomic libraries were generated and then later sequenced on Illumina HiSeq 2000 in a 2x150 paired end mode. These were then quality checked using Trimmomatic version 0.35, and the high-quality paired end reads from each of these samples were then assembled into contigs using MegaHit 1.1.3. These contigs were then evaluated using Quast 2.3 and then queried in PROCA and BLAST against NCBI database of non-redundant proteins and clusters of orthologous groups database using Megan 6 Ultimate Edition 6.20.5. Genome binning of assembled contigs carried out using MetaWrap. The completeness of these metagenome assembled genomes, otherwise known as MAGs, were evaluated using CheckM 1.1.2. MAGs were then annotated using PROCA and searched against NCBI non-redundant protein database and KEGG database. Phylogenetic trees were then constructed using GTBTK software and then generated using an online ITOL platform. A similar workflow structure has been made for the transcriptomics. 3 cDNA libraries created using red soil 0% treatment, red soil 0.5% treatment, and the black soil 0.5% treatment after 160 days. Next ultra-directional RNA library prep kit for Illumina was used on material from replicates of each microcosm described and was used to create a composite sample for cDNA library preparation. Yield was then determined using qubit double-stranded DNA HS assay kit and then quality evaluation by Experion DNA 12K analysis kit. The 3 cDNA libraries were sequenced in a paired end mode, 2x150 base pairs with an Illumina HiSeq 2000 instrument for a total of 20 gigabase pairs of metatranscriptomic raw sequence data from each sample. All raw sequences were quality trimmed using Trimmomatic 0.35. SortMe RNA was used on rRNA and tRNA samples to construct mRNA, and then these were blasted against NCBI non-redundant protein databases. The BLAST outputs were then taxonomically assigned by the lowest common ancestor method, which is a taxonomic method to define the deepest relationships as having all descendants, which was implemented by Megan 6 Ultimate Edition. Annotation of the mRNA reads was done using seed subsystems and KEGG categories. Bowtie 2 and Megan 6 Ultimate Edition 6.20.5 were used as pathway determiners and mapping of mRNA databases into Methanogen MAGs. Now, let's take a look at our results. Total DOC and DOM increases both in black soil by 80% and in red soil by 60%. As microbial communities worked concaminately to reduce total LDPE molecule mass to more and more bioavailable forms for later methanogenesis. As nanoplastic concentration increased, recalcitrant DOM molecules also increased. This is due to the uptake and subsequent reduction of labile DOM molecules. Continuing, as more LDPE molecules were degraded into more available forms, methane production increased significantly. Relative to the control, black soil methane production increased upwards of 10.1 times. For red soil, this number is smaller, at about 4.5 times increase in methane production. However, red soil would still continue having the higher total methane production compared to the black soil. This data corroborates with DOC and DOM results as they are metabolized by other microbial partners. MCRA copy numbers increases further compound these results, as the relative abundance of these genes indicates a significant and yet positive correlation between methanogen abundance and methane production. Fatty acid profiles exhibit a 30-day peak. This is expected due to LDPE naturally having hydrolytically degraded around 30 days. What these graphs indicate, more importantly, is that metabolite concentration in all samples reduced significantly at the 160-day sampling point. This may point to long-lasting effects in this regard to be insignificant with the given sample size. In the real world, however, plastic is continuously being introduced into the environment, signifying that recovery to an original state may be possible, but unlikely given the nature of plastic usage. Shifts in mag-related abundance positively correlate to methane production and DOC content. Overall, 36 soil samples underwent shotgun metagenomic sequencing and produced over 593 gigabytes of Illumina sequence data. 1,190 mags were assembled, however, only 391 of these were determined to be of high quality. Of the bacterial mags, only 45 had a significant and positive correlation with DOC content brought by LDPE degradation. Centrofomonaceae was the most prevalent, having 21 mags, with the other most prominent bacterial mags were Lentimicrobiaceae with 6 and Ignavibacteriaceae with 4. 38 high-quality methanogen mags were identified where more variety appears. Methanocella with 17 total mags, Methanobacteriaceae with 11, and Methanotrichaceae with 3. Of these methanogen mags, only 12 had relative abundance that were positively correlated with methane production, most predominantly of Methanocella with 9 and Methanobacteria with 3. Both soil samples observed these correlations between metagenomic abundance of Centrofomonaceae and Methanocelaceae mags, with increasing nanoplastic concentration. Correlated abundances were detected via hydrogenase genes in Centrofomonaceae. Centrophomonous species are known as keystone taxon for the anaerobic oxidation of fatty acids via beta oxidation. The study continues, showing that this is an essential component for many organisms, as it is reliant on a hydrogenotrophic methanogen for thermodynamic reasons. As Methanocelaceae specializes in low hydrogen conditions, this allows itself to become the dominant methanogen in later stages of the experiment when hydrogen conditions reach a low point. Enrichment of both of these organisms show a syntrophic oxidation of short-chain fatty acids and long-chain fatty acids, and the requirement for a hydrogenotrophic methanogenic partner. Relative abundance of methanogens to bacteria increased with nanoplastic concentration and incubation time. Overall, the abundance ratio shifts in favor of organisms with the ability to metabolize byproducts near the end of the experiment, as well as the organisms specialized in low DOM and hydrogen conditions, which happen to include several of the methanogenic taxa that were described in the previous slide. Shifts in relative metagenomic abundance in methanogenic pathways indicates a buildup of recalcitrant molecules where partnerships with other organisms cannot make up the difference in thermodynamic energy expenditure. Black soil, in particular, with 5% nanoplastic concentration, had the greatest methanogen to bacteria abundance ratio, whereas ratios between 0.5% and 5% in the red samples did not differ that much. Now to move on to our discussion. We understand that anthropogenic plastic waste has been shown to exacerbate methane production in the article. We could still be severely underestimating the true rate of global greenhouse gas emissions and also the amount of gas emissions that are stuck in, say, these underwater rice paddies as they are in Antarctic ice sheets and Arctic ice sheets. We don't truly know how these conditions exist in nature. For example, within waterlogged rice paddies themselves, the rhizosphere provides an oxygenated gradient that does not exist in the samples of this study. Given that methanogenesis is primarily an anaerobic process, this may not completely reflect the full picture. However, we must keep in mind that things are not as simple as they seem. So, to conclude our research, plastic waste is known to negatively affect a variety of organisms, animals, plants, fungi, etc. These waste particles are often degraded by ultraviolet light, water, and other chemical interactions. These plastic particles that are degraded eventually can reach sizes in the micrometer and nanometer range, having many unknown implications on microbial life. Methanogens in particular exhibit increased methane production in conjunction with partner organisms. The broader implications on greenhouse gases emissions may show an underestimation of our current understanding. And now some questions for you, the viewer. What are other anthropogenic microbial interactions that have global or regional impacts? How has your understanding of archaea and their many unique niches changed over time? Film mulching is a globally used technique for a wide variety of different crops. What are other farming techniques that might have microbial consequences? And what other plastics have environmental impacts and what makes them unique?